I'll just do the normal reminders since it's being recorded. So if anybody comes, can see. Uh, sort of a new reminder. If you have a bunch of absences, and some of you already do, you can still go back and watch the old videos. So like, let's say you have an absence from, I'm looking at somebody specifically just because it's in front of me. You have an absence from last Wednesday, right? February 1st. You weren't here, never sent me an email. You can still go back where I've posted those announcements on Google Classroom and watch the video and follow the directions. You only get half credit because you're late, but half credit's better than zero. So if you have zeros for attendance, I recommend doing that because again, attendance is graded. It's a good time to remind you guys too. It's actually on purpose. It's not supposed to make things harder. It's almost like free points in a way, right? So you might be bad at grading or bad at exams, or you might be bad at labs, but you get points just for showing up, or in this case, just for watching videos if you missed it. Yeah, so that's it for attendance reminders. Any questions about attendance reminders? What else? Lab. So if you haven't done last week's lab or this week's pre-lab, make sure you get it done because like I always say, as long as it's done before I get around to grading it, I consider that on time. And I haven't even started grading last week's lab. So, hint, hint, if you haven't done last week's lab. Also, I got a good email from one of you. I know it seems confusing, but you just never need to hit the submit button on the labs. So, and I'll sometimes use the wrong verbs and say you need to submit your labs. But when I say that, what I mean is you need to make sure you have them done. I only say submit labs because I'm thinking in my Bio 108 class, which is online, where they do need to hit a submit button. Yes, sir.
So do you mean like on Sundays as well? Like actually
always, yeah.
In the lab, not turning in the lab?
You're never hitting the submit button. That's okay. And speaking of which, and the person who sent me the good email reminded me of that. It's not a big deal. It makes things easier on my end and your end if you don't hit the submit button. But if you do hit the submit button, there's a way around it. It's just more.
Oops. Is that why it expires my QE right after I submit?
Probably, yeah.
That makes a lot of sense now.
Yeah, that's professors who want to be like, you better turn it in on time, otherwise it's late, right? Because then I'd be like, I could see exactly when you hit the submit button, but I'm not trying to do that. If I'm late to grade something, then you should be late. You should be allowed to be a little bit late to turn it in. This is what I see, at least for this class. Not like the online class. That's a different story.
So how do you fix it?
I don't know. That's a good question. Maybe if anybody knows, maybe we can meet online and make a little video to show how to do it, so I can show it to other students, and I give you a little extra credit for doing that. But good questions. All right, and it is 8, so I'll go back to attendance. Oh, we only have one new person. Anyway, Massey's here. All right, yeah, so anyway, turn those in. And again, like I said, don't hit the submit button, but if you do, it's not a big deal. We can work around it. I'm just trying to make things easier for everybody. For independent work, you should have about, 21.6 points, somewhere around there. So, if you don't, you should work on that. And if you need help, let me know. So any questions about anything? All right. Let's get started since it is 8:01. We're going to jump back into chapter 3. For attendance, I'm changing it up a little bit today. For those of you in person, this won't matter for you. If you're online, you're still doing attendance words, except it's going to be a little bit trickier today. Hold on, and I'll explain how. Okay, so today, instead of saying, here's the attendance word, I'm not going to say anything. I'm just going to circle it. So pay attention with your eyeballs. So when you see me circle a word, then that is the attendance word. So if you're in person and want to get the extra credit, you know what to do. If you're online, you need to send me those words in an email before 9 a.m. And if you're watching the video as opposed to being in the live stream, you need to take a screenshot of the circled word. So basically everything's the same. So to remind you, it's an in-person. And you have a computer in front of you, your brain's either going to do something fun or do something that you need to do, right? Productive or fun, but sometimes it's hard to focus on what you need to focus on. So anyway, let's get started with words and with the lecture. So yeah, we've talked about carbohydrates. You had to know the monomer and the polymers and the different types of carbohydrates. We talked about lipids. You needed to know the two different types of lipids we talked about and the difference between saturated and unsaturated fat and the building blocks of lipids, which are not necessarily monomers because they're not repeating the same parts. Now we left off with proteins. And I think, if I remember correctly, I already told you this, and this is what you need to know for the exam. Well, no, I don't think I have told you this yet. You should know this. This will be important, not just for this. Oops, I'm not going to circle anything. I'm not circling, just an arrow. Yeah, proteins. They're made up of amino acids. So those are the monomers. You need to know that monomers are the amino acids. And on top of that, unlike the other ones that I was teaching you, where you just needed to know the name of the monomers, now I'm also telling you, not only do you need to know that the monomers are called amino acids, but you need to know that there's 20 of them for living organisms, which is one of those ways that we're all related in a sense, right? All living things, whether it be a bacteria, snake, or a human, or a poisonous plant, whatever, you all have 20 amino acids that make up your proteins. So I also put a little star next to that because there are other amino acids that aren't like necessarily common to all living things. So you can look that up if you want. Also, if you're more interested in humans, you can look up which amino acids humans could create and which ones we need to eat in our diet. So anyway, any questions about what amino acids are in general? All right, so here's something else new that I'm teaching you because you needed to know that the monomer for carbohydrates are monosaccharides, right? You needed to know what lipids are made of. But this is the first time we're diving deeper into the actual monomer, right? So you weren't required to know what a sugar is made up of, right? You just needed to know sugars make up carbohydrates. Here, I'm telling you, you need to know the 4 parts of the amino acid. And I'm never going to show you this picture and like tell you, which part's the amino group, which part's the carboxyl group, which part's the side chain. I'm not going to do that. But you do need to know the 4 parts, right? So every single amino acid, all 20 of them, they all have this basic form, and you need to know it. The amino group, what's a central carbon, and as you know, carbon bonds 4 times. That's attached to an amino group, a hydrogen, a carboxyl group, and the side chain. So you need to know those 4 parts. And it's the side chain that defines it, if you will, right? So no matter what, all amino acids they have the same exact amino group, same exact hydrogen, of course, it's a hydrogen, the same exact carboxyl group. The only thing that's different is the side chain. They all have a different side chain, and that's what defines what they are. So if I say it's a tryptophan, that means it has a certain side chain that makes it a tryptophan. We'll get into more details for that later. Anyway, any questions so far? Proteins is a lot of information that I'm giving you. But we talk about protein. Move forward. Like this is said, except now so you can read it, right? So again, the amino acid is a central carbon bonded to 4 things, as carbon does. The 3 parts that are all the same for all amino acids is the carboxyl group, the amino group, the hydrogen atom, right? All that stuff I just said, there it is in writing. But the variable component is the side chain. And again, that's the thing that defines the amino acid, if you will, that makes it one amino acid different than the other. I'll go ahead and remind you guys, it's the last chapter, the first exam. So once we finish this chapter, we need to do our first exam. So start thinking about that for a little bit, because mostly I'm going to leave it up to you, not completely, but when the time comes, I'll say, all right, we can have the exam on this day, or this day, and then I'll ask you to give me some feedback. So start thinking, do you want it on a Monday, Wednesday, Friday? Do you want it directly after we finish this, or do you want some time to let it marinate and then jump into it? Again, there's just another picture. It's the same thing, a bit different, but still, amino group, hydrogen, carboxyl group, side chain. Same thing, different look. Again, this is everything I've said, but now it's in writing. Each amino acid has a unique side chain that gives it its special chemical properties. Actually, I haven't even mentioned that yet. So yes, that's what defines it, but it also gives it its special chemical properties. Because we talked about that a little bit earlier when we talked about side chains and functional groups, right? Some can be polar versus non-polar, which means they don't mix with each other, or hydrophobic versus hydrophilic, charged or not charged, positive, negative, all these things. Yet another picture. And of course, you don't need to know the difference between leucine and serine. These are just two examples. But even though you guys aren't chemists, you can just look at this and say, well, that's a different size and shape than that. So therefore, it probably has a slightly different function, and you would be correct. Because again, like I keep saying, when we talk about molecules, a lot of this, especially with proteins, is how they fit with each other. And you can see just by looking, because that is the shape of it, this thing would fit into something different than this thing, right? Here we have like a triangle looking thing. There we have a rectangle looking thing. And yes, they are different. And also, look, this one happens to be hydrophobic. Not that you need to know that. This one happens to be hydrophilic. If those two things were next to each other, they would repel each other, right? You couldn't mix them. Meanwhile, if you put this thing in water, it would repel from the water. If you put this thing in water, it would be attracted to the water, right? So all these different things that help it shape, right? Help it form its shape. And that'll be very important here in a few slides. So again, I'll remind you, the leucine, the serine, those are just examples. I'm not going to ask you, which one's hydrophobic, leucine or serine? This is just an example. So any questions about those examples? All right. What do we call it when we bring this together? As I keep saying throughout this chapter, when you bring them together, they are called dehydration reactions. But that would be too easy. So your book, biology, they just want to make things a little bit harder for you. So yes, they are still called dehydration. But we're going to get specific. When we're talking about proteins, they happen to be called peptide bonds. I'm going to put an X through this, only indicating that I'm not necessarily going to ask you about peptide bonds, but that will help you a little bit later. Actually, right now. Because, as I already told you, and you need to know this, the monomer for proteins, those are called amino acids. What are the polymers called? They're called polypeptides. So you need to know that. So that's the only reason I even bring up the fact that they're bonded together with peptide bonds. Not because I'm going to ask you, hence the X, but to help you remember that the polymer is called polypeptides. They're called polypeptides. So confusing, right? Carbohydrates are nice and simple. Monosaccharide, polysaccharide. It's all right there in the name. Here, we're like amino acid. In my opinion, if I could rename things, I'd call it a polyamino acid. That just makes sense. But again, that'd be too easy. We got to make things harder for students. So the monomer is amino acid. The polymer is polypeptide. And then it gets even trickier. Unlike any of the other stuff we talked about, this is still not even a protein yet. So it's just a long, just like a polysaccharide is a long string of glucoses, for example, right? So polypeptide, again, it's a long string of amino acids, but it's not yet a protein. So does anybody know what this is in general? Anybody guesses what that picture is? I can think of at least, like if I had to guess, I would guess, I would know, two different things. A basket, I could see that, but you're way back there. So yeah, but if you're a little bit closer, can you tell? Yes, I can see that. Okay. That sounds like more fun. What?
A quilt, like a handwood quilt.
Yeah, like a crocheted blanket.
Yeah.
That's my guess. Okay. I don't even remember what it is, but that would go along with what I'm trying to get at. So yeah, it looks like a quilt. Anybody, or a crocheted blanket. Anybody else see something different? I don't know. Maybe like a hat or a scarf. Anyway, can we all agree, except you way back there, maybe you can't see it, but can we all agree that's something made of yarn? Can you see that? All right. So let's just go ahead and assume that it's a crocheted blanket, right? So we'll call that a blanket, right? But if someone else were to say, if I say, all right, what is this? And she says, that's yarn. Is she incorrect? No. All right. Now, if I were just to hold up a bunch of yarn and I said, here's a blanket, am I correct? If it's just a long string of yarn, it's not a blanket yet, right? It's not a blanket until you've done your little magic with the crochet things and done that, right? So it's yarn until you put it into its shape. Then, while it is still yarn, it is considered, you know, a scarf or a blanket or whatever it may be, or even like a piece of paper. If I hold up a paper airplane and say, "What is this?" You guys would say it's a paper airplane, and then I would say, "But is this also not a sheet of paper?" So yes, it's also a sheet of paper. But if I were to just show you a sheet of paper that hasn't been folded, so this is a paper airplane, you'd be like, "No, that's not a paper airplane yet. That's just a piece of paper." So all that makes sense, because that's the relationship between a polypeptide and a protein. A polypeptide is nothing but a string of amino acids. It's not considered a protein until it folds and reaches its final shape. That'll be very important later in the semester. All right. So are there any questions about this slide? About peptides, polypeptides, peptide bonds, the relationship between peptides and... Okay. And for the most part, I'm not even going to be picky. So in questions and in exams, if you happen to call a protein a polypeptide or a polypeptide a protein, usually you're not even going to lose points, unless the question is specifically asking you about that. Like if there's a question that specifically is looking for the difference between a polypeptide and a protein, then you need to know it. But other than that, I'm not going to be picky. I'll even make the mistake sometimes. I'll say polypeptide when I should say protein, or vice versa. But anyway, let's move forward. Again, this is just everything we said, but now it's in pictures. Here we have one amino acid mixing with another. It's a dehydration reaction. It's forming a covalent bond, which again, we have to make more complicated and call it a peptide bond. It's just a covalent bond. It just happens to be between amino acids, so we call it a peptide bond. Just any questions about that? Okay, so your body has thousands of proteins, and that's a good independent work topic if you want to look it up. Exactly how many do we have? And that number changes as technology gets better and people do research, but it is accurate to say there's thousands. But if you want to look it up, how many exactly do humans have? Or what about any other organism that you're interested in? If you like those invasive species, what are those things called? Cats. If you like cats, you can look that up. Anyway, so there's thousands of proteins, but there's only 20 amino acids. So hopefully that begs the question, well, how is that possible? How do we have thousands of different proteins, but we only have 20 different amino acids? And your book uses a good analogy, and we're talking about the English language, right? We have 26 letters in the English language, but we have all these different words, right? Because we put those letters together in different combinations. And it's the same thing with protein, right? So yes, we only have 20 amino acids, but we put them together in different combinations, and that's how we get all these different proteins. And again, this is going to be very important later in the semester. So you may have noticed it's taken me a lot longer to get through the protein section than the other stuff, because this is more important for the rest of the semester. Anyway, any questions so far?
You just reiterate how it breaks down.
Yeah, so, you know, there's thousands of proteins in our bodies. So the question is, how do we have thousands of them when we only have 20 amino acids to work from? And it's just because we put them together in different combinations. And your book uses the English language as an example. So we have, you know, millions of different words in English. How do we do that with only 26 letters? It's just different combinations of how we put them together. Anybody else? Yeah, when I made this PowerPoint a long time ago, that's how many words there were in the English language when I looked it up. I'm sure it's different now. I would say look that up for independent work, but it's not an English class, so it wouldn't be any use. Anyway, if you download this PowerPoint, you can watch this animation. This shows the primary structure of proteins, and the primary structure is really just, all right, which amino acids are attached to which, right? What is the order? What is the sequence of amino acids? And it is that sequence that determines the three-dimensional structure, right? Because I keep saying that it's not a protein until it folds up and twists and finds its final shape. And it's the sequence that determines that. And this is a picture that's not from your book, and it's just a good example of how it's the sequence that determines it. The fact that we have this amino acid here that has a sulfur, and the same one here that has a sulfur, and they attract each other, and there's a covalent bond there. The fact that those two amino acids happen to be where they are makes this thing bend the way it does, because they're attracted to each other. So that's what I mean. That's what this means when I said it is the the sequence of amino acids that determines the structure. And a really, really, really important thing outside of this chapter 2, moving forward, this is a very important concept. It is the three-dimensional structure of the protein that enables it to carry out its specific function. The proteins are all about the shape. There's a question, I don't even remember how it's worded, but you could just write it down that way if you want. Proteins are all about the shape. It's not about the size, the weight, it's about the shape. It's like I keep hinting at, right? When molecules, at the molecular level, a lot of chemical reactions happen because of how molecules fit together, like puzzle pieces. So yeah, it's a very important bullet point right there. Proteins are their function is very much dependent on the shape, and that's basically what the second other bullet point gets at, right? So nearly all proteins work by recognizing and binding to some other molecule. Which again, that's just reinforcing this more important bullet point. It's the three-dimensional structure of the protein that allows it to carry out its specific function. All right, questions? Here's a picture actually from your book. So that last acid sequence of a lysosome. Just an example you need to know. But if you could see it, it might be hard for you back there. It shows you exactly which, amino acid is which. Of course, the only reason it's shaped like that, they have it all back and forth. It's just they're trying to get it on the screen. Really, it would be better if it was all straight because we're not concerned about the shape yet. This is just the sequence. But then, because of the sequence, again, that determines its three-dimensional shape. And those are just two different ways of looking at the three-dimensional shape. And it's all about the shape, right? The shape determines its properties. It determines how the protein behaves, if you will. So anyway, again, that's just an example. I'm not going to ask you about lysozymes. So any questions? All right. We're still talking about the shape here. If you have a slight change in the sequence, that can affect the protein's ability to function. If you change the sequence of amino acids, if you change one for another, or maybe add an extra or take one away, whatever, then obviously you're going to change its shape. And if you change its shape, you're going to change its function. And your book uses this great example, and we're going to use this example in so many chapters. When we talk about mutations, we're going to talk about this. When we talk about evolution, we're going to talk about this. But yeah, so this is one good example. So if you substitute one amino acid in the protein that makes up hemoglobin, that gives it a wrong shape, so to speak. The shape's off, so it doesn't do its job properly, and that causes what we call sickle cell disease. And I'll talk a little bit more about that on the next slide. But here's a good example. You can see, I don't know, you guys aren't chemists, so I don't expect you to be really familiar with what you're looking at, but if you can see it, it's a little bit hard. We have hydrogen bonds here, right? So this amino acid is hydrogen bonded to that one. And this amino acid is hydrogen bonded to that one. That's what those little dots mean. And as you know, and as you do need to know, hydrogen bonds are the weakest of the bonds, right? So if you were to change one of these amino acids to where that one, for example, wouldn't be hydrogen bonded to that one anymore, then it would lose that thing holding it together, right? It would be no longer a spiral. And of course, we don't get into too much detail, but the point here is, again, if you change that one amino acid, you're going to change the shape. And if you change the shape, you're going to change the function. So let's talk more about the hemoglobin example when we look at it. So this is what a normal hemoglobin looks like at the top. This is what one with sickle cell looks like at the bottom. Of course, when you just look at the polypeptide itself, unless you have a trained eye, that might not look much different. But it is. And you can see how the blood cells look a lot different. You don't need a trained eye to see that. That's a normal red blood cell. That's a sickle red blood cell. And when we talked about form and function, remember we talked about blood cells. We said, they have that shape, that broad, flat shape to help diffuse gases. Well, you mess up the shape, you mess up the function. And all this because of this one amino acid, right? So we substituted one for another. Out of all these amino acids, we just substituted one. And from that, we get this whole different shaped protein. And from that, you have a whole disease. If you want to look more, these for the, who has it, things like that. Any questions? Mm-hmm. And like I said, we'll use sickle cell disease as an example in other chapters. All right. Again, this is everything I just said, but now it's in writing, so you can read it too. One difference is enough to cause a protein to fold into a different shape. And sometimes that causes diseases. And your book actually points out a certain type of misfolded proteins. Those are called prions. And they cause nervous system disorders. And I'll give you some examples on the next slide. If I ask anything, someone put a big old question mark next to prion or over prions, because I don't think I might not ask anything about it. But if I do, the only thing you need to know about prions is they are misfolded proteins. So yes, they affect the nervous system. There might be questions about that in the study guide. I don't remember. But for you, for the exam, if anything, you just need to know the prions are misfolded proteins. I would leave it at that, but I think they're actually the brain. You can see these normal shaped proteins and these misshaped proteins, and the way it's so bad is when those misshaped proteins come in contact with other proteins that are normal, then they make those things misfold. Then they kind of cluster up, and basically you get holes in your head, and your whole brain's like a sponge. Hence the name, for example, bovine spongiform encephalopathy, BSE, otherwise known as mad cow disease. So here you have three examples. Mad cow disease, Kuru, and fatal weight loss in deer, elk, and moose. So if you want to, for independent work, look up some other ones. Or look up Kuru. Is a really interesting history on Kuru. I mean, I'm sure you can tell what kind of skulls these are. Any guesses? Human skull, yeah. So most of the way, the big way that Kuru was passed around was through cannibalism. People eating other people's heads, brains. So, interesting history if you're into it. So any questions about this? Again, just examples. I'm not going to ask you any questions about that. If anything, just know that prions are misfolded proteins. Another thing I didn't mention, but a protein shape is sensitive to its environment. And again, it's all about the shape, right? If we talk about proteins, the shape is so important because that's how they work at the molecular level. They need to fit together with other molecules. So this is very important. Hopefully you can see this. You can see these dotted lines. Like I mentioned before, those are hydrogen bonds. Not that you need to recognize that for an exam. But again, what you do need to know for the exam is that hydrogen bonds are the weakest of bonds. So what happens when you apply some heat to this? Well, eventually there's enough energy for those bonds break. And those are pretty weak bonds, right? They're the weakest. So what could happen is if you apply some heat to this, as one example, this whole thing is going to unravel. It's no longer going to be a spiral. It's going to be a long string, right? So then it's going to lose its shape. Therefore, it's not going to be able to do its job. So that's just one example of how temperature works. pH, right? If we say something is acidic, what does that mean? There's a bunch of what in the solution? If something is acidic. And this is something else you need to know for the exam. All right. I'll remind you. H plus ions, right? If something is acidic, there's a lot of H plus ions. And then obviously that's positive. Now here we have a hydrogen bond again, right? So this oxygen is slightly negative, right? Because the electrons like to hang out there. This hydrogen is slightly positive. So what's going to happen? Yeah, those things are attracted to each other slightly because of hydrogen bonds. But this thing is fully positive. So what's going to happen instead is that oxygen is going to bond to that hydrogen, right? If this is in an acidic environment. So that's going to disrupt those hydrogen bonds. So therefore, the thing's going to unravel. It's going to lose its shape. Therefore, it's going to lose its function. That's just one example. I'm not going to ask you the mechanisms of how that works. But hopefully you can see that, right? If you have something positive in there, it's going to disrupt those hydrogen bonds. Same thing if it was really basic. So if it had a bunch of, this is a good time to remind you, if it's basic, it would have a bunch of OH minus, right? That's what we mean by basic. That's so hard to read. OH minus. But again, that's how it would work. This hydrogen that's slightly positive would be attracted more to that OH minus. It would disrupt the bonds, the hydrogen bonds. It would unravel. It would change its shape, and it would change its function. And a lot of times, that's how people can die from being overheated. They call it denaturing. Your proteins just melt. Anyway, any questions about that? Long story short, for the exam, a protein shape is sensitive to its environment, so heat, pH, stuff like that. Awesome. So that brings us to the last of the 4 categories of biological molecules. And this is another really important one. So we talked a little bit about carbs later in the semester, a little bit about lipids later in the semester, a lot about proteins, and a lot about nucleic acids. So this one is pretty much just as important as the last one. All right, what do nucleic acids do? You should know this for the exam. Not like I expect you to know it right now, but for the exam, you should know that nucleic acids store information and they provide instructions for building proteins. So nucleic acids, that's the big one in my opinion. As far as the stuff we talk about in this this semester, that's the important one. Nucleic acids provide instructions for building proteins. That looks like garbage, but there it is. Yeah, nucleic acids store information, provide instructions for building proteins. There's 2 types of nucleic acids. You need to know them, and you're going to need to know the differences between them. There's going to be questions about it on this chapter, or this exam, and then later in the semester when we actually talk about the whole chapter about nucleic acids. So then this is going to come back and you need to know it then too. So the 2 types of nucleic acids are DNA and RNA. You need to know that. Yes, technically DNA is called deoxyribonucleic acid. For now, you can just know it as DNA. Yes, technically RNA is called ribonucleic acid. For now, you can just think of it as RNA. Later, when we have the whole chapter where we get into this, then the name will be important. But for now, just remember DNA and RNA. And we're going to talk about both of them. So for now, are there any questions? All right. That brings up another term that you need to know. And this is going to be very, very important later. Slightly important for this first exam, but now we're just scratching the surface. I might not even ask you about what a gene is on this first exam because we're going to devote so much time to it later. So I'm going to put a question mark just to indicate that I might or might not ask what a gene is on this first exam, but it'll definitely come back later. Anyway, what is a gene? It is a unit of inheritance encoded in a specific stretch of DNA that programs the amino acid sequence of a polypeptide, blah, 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 blah. A gene is instructions on how to build a protein. There you go. That's how I would write it. You can write it any way you want. I don't want you to memorize it verbatim, especially if those words don't mean anything to you. I'm telling you, a gene is a set of instructions on how to build a protein. And yes, those instructions are written in your DNA, right? It's a set of instructions on how, whoops, I'm not circling that. I almost did. Just drawing your attention to it, DNA. Didn't circle it. Anyway, that's what a gene is. A piece of DNA that gives you instructions on how to build a protein, a polypeptide. I'm going to definitely put it next to this because this is really deep, and we're going to get into that later. Later, I'm definitely going to ask you about this, but for now, I'm just going to introduce you to it. Exam 1, I'm not going to ask you about this, but I will definitely ask you this in later exams. The instructions that I'm talking about on how to build a protein, they are written in a chemical code. So all the A's and T's and C's and G's that you're going to learn about later that make up the DNA, that's the code. And those things have to be translated, going from the nucleic acid language to a protein language. And we're going to learn all about that later in the semester. So again, for the exam, if anything on this slide, you need to know what a gene is, and maybe not on this exam, definitely on another exam. And a gene is a set of instructions on how to build a polypeptide. Any questions about that? All right. There's a basic visualization of what we're talking about. So there's a long strand of DNA. Of course, we're only seeing a part of it. And then a small part of that strand of DNA is a gene. So if we could see, there's a code written there, but we can't see that code. That is then changed into RNA, and you're going to learn about that later. And then that RNA is then changed into amino acids. or I guess a more accurate way of saying that, even though it doesn't matter for this exam, but it will later. The information written in this is basically copied into this, and then that information is used to dictate how to build this. And again, that will be important later in the semester, but not for exam 1. All right, here we go. Again, you need to know this. The monomers, the polymers, right? So when we're talking about nucleic acids, you need to know that the monomers are called nucleotides. So now we've covered all the monomers. The monomers of carbohydrates are called monosaccharides. The monomers of proteins are called amino acids. And the monomers of nucleic acids are called nucleotides. In the same way you needed to know what the amino acids, how they were generally built, same with this. You also need to know how the nucleotides are built. So again, the amino acids had 4 parts, right? Here, the nucleotides have 3 parts. You need to know these 3 parts. A 5-carbon sugar, a phosphate group, and a nitrogen-containing base. And of course, I bolded the words you really need to know. So if you wanted to shorthand that, for your purposes, you could just memorize sugar, phosphate, and base. I don't care that it's a 5-carbon sugar. That does not matter for you guys. That won't matter. There is a difference in the chemistry, but for you guys, that doesn't come into play. Phosphate group, yeah, who cares? It's a phosphate as far as you guys are concerned. And again, it's the base, nitrogen-containing base. So again, sugar, phosphate, base. I'm trying to really draw some parallels here between these and amino acids. So remember, the amino acids had the 4 parts, right? And all three of those parts were the same. It was the side chain that defined the amino acid, right? That made this amino acid different from that one, so on and so forth. Well, with the nucleotides, the part that changes is the base, right? So the sugar, the phosphate, those are the same. It's the base that defines the nucleotide. There's the sugar, there's the phosphate group, and there's the base. Again, all these parts are the same. The sugar and the phosphate, that's the same. And I'm not circling that for any reason other than to show you that. That is not an attendance word. It's a big squiggly mess. Me showing you the sugar and the phosphate. And then, yes, there's the base. That's the part that varies. That's the part that defines the nucleotide. And we're going to talk a little bit more about this, so I'm not going to take questions yet. There's a picture that is from your book. Again, there's your sugar, there's your phosphate, there's your base. And again, it's the base. That's the part that varies. That's the part that defines it. It comes in 4 varieties. So unlike amino acids that come in 20 varieties, these come in 4. So because there's only 4 and there's not 20 of them, you need to know them. A, G, C, and T. You need to know that. So again, you need to know nucleotides are the monomers. You need to know that they're made of 3 parts. You need to know the 3 parts are phosphate, sugar, and base. You need to know that the base is the part that varies, and that the 4 different types of bases, for DNA anyway, are A, G, T, and C for DNA. You need to know that. And they have names, and we'll talk about the names later. But really, just the letters will work. The A, G, and the T, and the C, that works. Like, for example, here, it shows you right there thymine, but we just call it a T. And for a 100 level non-biology major course, the letters are just fine. Any questions about that? All right. Like I said, you can see it in writing. If we're talking about DNA, there's 4 different versions of the bases, unlike amino acids that have 20 different versions. There's A, G, T, and C, adenine, guanine, thymine, cytosine. There's other ones too, but again, not for DNA or RNA. So yes, just memorize those 4, A, G, T, and C. Any questions? All right, that's what they look like. Not that I'm going to ask you. I'm not going to show you a picture of adenine and say, is that A, G, T, or C, right? You don't need to recognize anything. I will point out, and this will be slightly important later, notice the A and the G have 2 rings, and the C and the T are single ring. That'll be slightly important later. You don't need to memorize which ones have 2 rings and which have 1 ring. Right? They have different sizes, different shapes, and that will be slightly important later. All right, so what is it that brings these nucleotides together? You might have guessed it. Hopefully you guessed it. Dehydration reactions, right? So like I keep saying, whenever we're bringing stuff together in this course, that's going to be a dehydration reaction. So that's what we use to link up those nucleotides, those monomers, which are the nucleotides. And what do we call the polymer? I'm finally getting to it because, as always, like I keep saying, you need to know the monomer, you need to know the polymer. They are called polynucleotides. And I just underlined that, didn't circle it. But yeah, polynucleotides, that's the polymer. That's why I like nucleic acids. The name, the nomenclature just makes sense, right? The monomer is a nucleotide. The polymer is a polynucleotide. Nucleic acids, mononucleotide, polynucleotide. It all fits together. It's not like those confusing proteins. Proteins, polypeptides, amino acids. Anyway, I'm not going to ask you this, but just so you know, when we're talking about which parts are bonded to which parts, just for your own information, it's the sugar of one the sugar of one nucleotide. It's a covalent bonded to the phosphate or another. And that's where we get this term, the sugar phosphate backbone. So remember, there's 3 parts to a nucleotide: the sugar, the phosphate, and the base. So basically what I'm telling you here is it's the sugar and the phosphate that really matter. It's the sugar and the phosphate that are holding these things together. The bases just kind of flapping out there in the wind, so to speak. And that'll also be important later, but not for this first exam. So if anything, I probably, again, I'll put it next to that because I'm probably not going to go this deep. But if I do ask, you need to know it's the sugar and the phosphate that are covalently bonded. That's what's keeping those things together. The bases are not involved in keeping that string of nucleotides together. They do have a function, obviously, and we'll talk about that later. But they don't function by keeping that string together. DNA is a double-stranded molecule, meaning that basically you have 2 polynucleotides right next to each other, and they're hydrogen bonded to each other. They twist around each other, hence the double helix. But for this exam, if anything, this is all you need to know from this bullet point anyway. DNA is double-stranded. Because, again, to throw back what I was talking about earlier, there's 2 different types of nucleic acids, DNA and RNA. And you need to know the differences and the similarities between both of them. So I'm telling you right now, the first difference is DNA is double-stranded. So what is it that keeps these 2 strands of polynucleotides together? It's those bases, right? So a single polynucleotide is kept together by that sugar-phosphate backbone, right? Those covalent bonds between the sugars and the phosphate. That's what keeps, I don't know, let me try to draw it. That's what keeps this single strand, of course, man, I hate this drawing thing. Forget those little things off to the side, but there's a single strand of DNA, right? The thing that's keeping those polypeptides together, those polynucleotides together, covalent bonds. Then you have this strand, and it's being kept together with covalent bonds between the nucleotides. So what's keeping those 2 strands together with each other? And those are hydrogen bonds, right? So there's hydrogen bonds between the 2 strands, and it's between the bases of the strands. But after going through all that, I'm going to put an X through this, indicating that I'm not going to ask you this on the first exam. Later, when we really get into nucleic acids again, you will need to know that. For now, it's just an introduction. I'll put a question mark next to this because I don't know if I'm going to ask it on this exam. It'll definitely be on the other exam. But you should know that A's go with T's and C's go to G's. So if there's an A on this strand and there was a T on this strand, they would go together. They would be attracted to each other. If there was a G on this strand, on the other side, there would be a C. So those are the things that go together. A's go with T's and C's go to G's. All right, let's see. Before I go to the next slide, I know that looks really wonky. I hate the way this thing draws, but that is a circle. Yeah, so the next slide I'm going to show you does a better job of visualizing what we're talking about. I tried to draw it. As you can see, it's really wonky. So let's look at this next slide, see what it's supposed to look like from your book. There we go. So not only do we have the 2 different strands, but you can see the twisted together. That's what makes it double helix. And again, this strand is covalently bonded to itself, right? That's what keeps that strand together. This strand is covalently bonded. That's what keeps that strand together. But what keeps this strand Bonded to this strand, it's the hydrogen bonds between the bases. And again, that'll be important later, because we know that covalent bonds are the strongest bonds. We know that hydrogen bonds are the weakest bonds. That's a little prequel. That's going to be important later. But for now, it's not as important. So again, there's differences and similarities between DNA and RNA, and you need to know them. Both DNA and RNA are polymers and nucleotides, right? So whether you're talking about DNA or RNA, the monomer is are nucleotides, and the polymer is a polynucleotide, regardless if you're talking about DNA or RNA. And no matter what, if you're looking at DNA or RNA, those monomers, those nucleotides, are made up of the same three parts, as far as we're concerned: sugar, phosphate, and a base. Now the sugar is different. We don't talk about that. The bases, there is one base that's different. We'll talk about that in a second. But so far, I just want to talk about how these things are the same. All right, same monomers, same polymers, same three parts that make up the monomer. Any questions about that before we start talking about the differences between the two? Of course, you can see them in that picture, but let's talk about it. Three differences. I'm going to put it next to the first one because I'm not going to ask you, but it might help you remember. The RNA sugar, like I just mentioned earlier, is different than the DNA sugar. Specifically, the RNA is ribose. DNA is deoxyribose. None of you are chemists. Only one of you is a biology major. And even then, for the biology major, that might not matter either. So that's why I put it next to it. You don't need to know that. For us, it's unimportant. It does help explain the different name, though, right? Ribonucleic acid versus deoxyribonucleic acid. Here's a big one. You definitely need to know this second bullet point. So DNA, remember, the bases for DNA are A, C, T, and G. RNA doesn't have a T. RNA has a U instead of a T. You need to know that for this exam. You'll need to know that for the future exam. RNA does not have a T. It has a U instead. And another one that you need to know for both exams, RNA is single-stranded, DNA is double-stranded. And this is all not true. Just some stuff that we're throwing out there and saying you need to know. So any questions about the similarities or the differences between RNA and DNA? All right. And then there's a picture that kind of shows the differences. So again, the phosphate group's exactly the same. The bases, RNA has A, C, and G, but it doesn't have a T. It has a U instead. So there's that. And again, it's a ribose instead of a deoxyribose, which for you guys doesn't matter, but there it is. That's a picture of a nitrogenous base for RNA. All right. Every chapter has this, the process of science. Every study guide, I think, has questions about it. So, read it. Let me know if you have questions. But as always, I'm going to skip past it. Actually kind of interesting, too. If you want to, you just scratch the surface when we talk about lactose intolerance. And if you want to look it up for independent work, it is pretty interesting, I think. I will point this out. This is not important for the semester, just interesting to me. The people who, anybody here lactose intolerant by any chance? Some of you? Okay. So that's actually normal. That's a normal thing for mammals and humans, is to be lactose intolerant, because technically, once you get past being a certain age, you're not supposed to have milk anymore, technically. But in some cultures, we've been able to eat lactose, and the reason is we all have the same gene, but then there's something different in our DNA. So we still have that gene to be able to eat lactose, but then there's something else farther down in the chromosome. Anyway, that's all I want to say about it. You can look it up for independent work if you want. That's it. So if you have lab, I'll see you at noon. Don't be late. Don't be late. Don't be late. And make sure you're wearing closed-toed shoes and pants. That's it. What? Oh, I'm not answered. Great question, but I'm not answered. No, do the pre-labs.
We're doing pH stuff now.
No, do the pre-labs. Do the pre-labs. You know what we're doing. Because you do the pre-lab. So yes, we're going to detect all these molecules that we talked about. We're going to detect them. If you could send me